Viral evolution. Viral evolution is a subfield of evolutionary biology and virology that is specifically concerned with the evolution of viruses. Viruses have short generation times, and many, in particular RNA viruses, have relatively high mutation rates, on the order of 1 point mutation or more per genome per round of replication. This elevated mutation rate, when combined with natural selection, allows viruses to quickly adapt to changes in their host environment. In addition, most viruses provide many offspring, so any mutated genes can be passed on to many offspring quickly. Although the chance of mutations and evolution can change depending on the type of virus, double-stranded DNA, double-stranded RNA, single-strand DNA, etc., viruses overall have high chances for mutations. Viral evolution is an important aspect of the epidemiology of viral diseases such as influenza, influenza virus, AIDS, HIV, and hepatitis, for example HCV. The rapidity of viral mutation also causes problems in the development of successful vaccines and antiviral drugs, as resistant mutations often appear within weeks or months after the beginning of a treatment. One of the main theoretical models applied to viral evolution is the quasi-species model, which defines a viral quasi-species as a group of closely related viral strains competing within an environment. Origins. Viruses are ancient. Studies at the molecular level have revealed relationships between viruses infecting organisms from each of the three domains of life, suggesting viral proteins that predate the divergence of life and thus infecting the last universal common ancestor. This indicates that some viruses emerged early in the evolution of life, and that they had probably arisen multiple times. It has been suggested that new groups of viruses have repeatedly emerged at all stages of evolution, often through the displacement of ancestral structural and genome replication genes. There are three classical hypotheses on the origins of viruses and how they evolved. Virologists are, however, in the process of re-evaluating all of these hypotheses. One of the problems for studying viral origins and evolution is the high rate of viral mutation, particularly the case in RNA retroviruses like HIV/AIDS. A recent study based on comparisons of viral protein folding structures, however, is offering some new evidence. Fold superfamilies, FSFs, are proteins that show similar folding structures independent of the actual sequence of amino acids, and have been found to show evidence of viral phylogeny. Thus viruses have been found to be capable of being divided into 4 FSFs, based upon the 3 realms of bacterioviruses, archaeoviruses, and eukaryoviruses, together with a 4th FSF that seems to indicate that it predated the separation of the 3 realms. The proteome of a virus, the viral proteome, still contains traces of ancient evolutionary history that can be studied today. The study of protein FSFs suggests the existence of ancient cellular lineages common to both cells and viruses before the appearance of the last universal cellular ancestor that gave rise to modern cells. Evolutionary pressure to reduce genome and particle size may have eventually reduced viral cells into modern viruses, whereas other coexisting cellular lineages eventually evolved into modern cells. Furthermore, the long genetic distance between RNA and DNA FSFs suggests that the RNA world hypothesis may have new experimental evidence, with a long intermediary period in the evolution of cellular life. A final exclusion of a hypothesis on the origin of viruses is difficult to make on Earth because viruses and cells interact with each other everywhere today, and very old rocks in which old traces of viruses could be found are probably rarely or no longer present on Earth. From an astrobiological point of view, it has therefore already been proposed that on celestial bodies such as Mars not only cells but also traces of former virions or viroids should be actively searched for. If only traces of virions but no cells are found on another celestial body, this would be a strong indication of the virus first hypothesis. Evolution. Viruses do not form fossils in the traditional sense, because they are much smaller than the finest colloidal fragments forming sedimentary rocks that fossilize plants and animals. However, the genomes of many organisms contain endogenous viral elements, EVs. These DNA sequences are the remnants of ancient virus genes and genomes that ancestrally invaded the host germline. For example, the genomes of most vertebrate species contain hundreds to thousands of sequences derived from ancient retroviruses. These sequences are a valuable source of retrospective evidence about the evolutionary history of viruses, and have given birth to the science of paleovirology. The evolutionary history of viruses can to some extent be inferred from analysis of contemporary viral genomes. The mutation rates for many viruses have been measured, and application of a molecular clock allows dates of divergence to be inferred. Viruses evolve through changes in their RNA, or DNA, some quite rapidly, and the best adapted mutants quickly outnumber their less fit counterparts. In this sense, their evolution is Darwinian. The way viruses reproduce in their host cells makes them particularly susceptible to the genetic changes that help to drive their evolution. The RNA viruses are especially prone to mutations. In host cells, there are mechanisms for correcting mistakes when DNA replicates, and these kick in whenever cells divide. These important mechanisms prevent potentially lethal mutations from being passed on to offspring. But these mechanisms do not work for RNA, and when an RNA virus replicates in its host cell, changes in their genes are occasionally introduced in error, some of which are lethal. One virus particle can produce millions of progeny viruses in just one cycle of replication, therefore the production of a few dead viruses is not a problem. Most mutations are silent and do not result in any obvious changes to the progeny viruses, but others confer advantages that increase the fitness of the viruses in the environment. These could be changes to the virus particles that disguise them so they are not identified by the cells of the immune system, or changes that make antiviral drugs less effective. Both of these changes occur frequently with HIV. Many viruses, for example, influenza A virus, can shuffle their genes with other viruses when two similar strains infect the same cell. This phenomenon is called genetic shift, and is often the cause of new and more virulent strains appearing. Other viruses change more slowly as mutations in their genes gradually accumulate over time, a process known as antigenic drift. Through these mechanisms, new viruses are constantly emerging and present a continuing challenge in attempts to control the diseases they cause. Most species of viruses are now known to have common ancestors, and although the virus-first hypothesis has yet to gain full acceptance, there is little doubt that the thousands of species of modern viruses have evolved from less numerous ancient ones. The Morbilliviruses, for example, are a group of closely related, but distinct viruses that infect a broad range of animals. The group includes measles virus, which infects humans and primates, canine distemper virus, which infects many animals including dogs, cats, bears, weasels and hyenas, rinderpest, which infected cattle and buffalo, and other viruses of seals, porpoises and dolphins. Although it is not possible to prove which of these rapidly evolving viruses is the earliest, for such a closely related group of viruses to be found in such diverse hosts suggests the possibility that their common ancestor is ancient. Transmission. Viruses have been able to continue their infectious existence due to evolution. Their rapid mutation rates and natural selection has given viruses the advantage to continue to spread. One way that viruses have been able to spread is with the evolution of virus transmission. The virus can find a new host through. There are also some ideas behind the idea that virulence, or the harm that the virus does on its host, depends on a few factors. These factors also have an effect on how the level of virulence will change over time. Viruses that transmit through vertical transmission, transmission to the offspring of the host, will evolve to have lower levels of virulence. Viruses that transmit through horizontal transmission, transmission between members of the same species that don't have a parent-child relationship, will usually evolve to have a higher virulence. Will usually evolve to